ਵਿਦਿਆਰਥੀਓ, ਕੀ ਤੁਸੀਂ ਜਾਣਦੇ ਹੋ ਕਿ ਲਿਵਿੰਗ ਸਿਸਟਮਸ ਨਾਨ ਲਿਵਿੰਗ ਐਟਮਸ ਅਤੇ ਮੋਲੀਕਿਊਲਸ ਤੋਂ ਬਣੇ ਹੁੰਦੇ ਹਨ ਜੋ ਕਿ ਵੱਖ-ਵੱਖ ਜਟਿਲ ਬਾਇਓਮੋਲੀਕਿਊਲਸ ਹਨ। ਇਹ ਕਾਰਬੋਹਾਈਡਰੇਟਸ, ਪ੍ਰੋਟੀਨਸ, ਨਿਊਕਲਿਕ ਐਸਿਡਸ ਅਤੇ ਲਿਪਿਡਸ ਹੋ ਸਕਦੇ ਹਨ। ਅੱਜ ਦੇ ਇਸ ਪਾਠ ਵਿੱਚ ਅਸੀਂ ਆਪਣਾ ਧਿਆਨ ਕਾਰਬੋਹਾਈਡਰੇਟਸ ਤੇ ਕੇਂਦਰਿਤ ਕਰਾਂਗੇ। ਕਾਰਬੋਹਾਈਡਰੇਟਸ ਕੀ ਹਨ? ਅਸੀਂ ਇਹਨਾਂ ਨੂੰ ਕਿਵੇਂ ਬਣਾ ਸਕਦੇ ਹਾਂ? ਅੱਜ ਦੀ ਇਸ ਕਲਾਸ ਵਿੱਚ ਅਸੀਂ ਕਾਰਬੋਹਾਈਡਰੇਟਸ ਨਾਲ ਸੰਬੰਧਿਤ ਸਾਰੇ ਮਿਸਟੀਰੀਅਸ ਫੈਕਟਸ ਦਾ ਖੁਲਾਸਾ ਕਰਾਂਗੇ। ਸਾਡਾ ਅੱਜ ਦਾ ਵਿਸ਼ਾ ਹੈ ਕਲਾਸੀਫਿਕੇਸ਼ਨ ਆਫ ਕਾਰਬੋਹਾਈਡਰੇਟਸ। ਟੌਪਿਕ ਸ਼ੁਰੂ ਕਰਨ ਤੋਂ ਪਹਿਲਾਂ ਅੱਜ ਦੀ ਕਲਾਸ ਦੇ ਲਰਨਿੰਗ ਆਬਜੈਕਟਿਵਸ ਤੇ ਨਜ਼ਰ ਮਾਰ ਲਈਏ।
Upon completion of this topic, learners will be able to define carbohydrates, classify carbohydrates, write preparation, properties and structures of glucose, draw cyclic structures of glucose and fructose, write structures of fructose, sucrose, starch and cellulose. Write importance of carbohydrates.
Carbohydrates refer to a large number of non-nitrogenous compounds. Initially, the name carbohydrate, meaning hydrate of carbon, was given to a class of compound containing only carbon, hydrogen and oxygen. The hydrogen and oxygen being present in the same ratio as in water. ਇਸ ਲਈ ਵਿਦਿਆਰਥੀਓ, ਅਸੀਂ ਕਾਰਬੋਹਾਈਡਰੇਟਸ ਨੂੰ ਇੱਕ ਜਨਰਲ ਫਾਰਮੂਲਾ ਰਾਹੀਂ ਦਰਸਾਉਂਦੇ ਸੀ। ਇਹ ਸੀ CxH2OY, ਪਰ ਇਹ ਸਾਰੀਆਂ ਕਿਸਮਾਂ ਵਿੱਚ ਲਾਗੂ ਨਹੀਂ ਹੁੰਦਾ। ਇਸ ਨੂੰ ਸਪਸ਼ਟ ਕਰਨ ਲਈ ਆਓ ਕੁਝ ਉਦਾਹਰਨਾਂ ਲਈਏ। In case of acetic acid CH3COOH, this general formula is applicable, but it is not a carbohydrate. Rhamnose C6H12O5 is a carbohydrate, but it does not follow this rule. Thus, carbohydrates are defined as follows. Chemically, carbohydrates may be defined as optically active polyhydroxyaldehydes or polyhydroxyketones, or the compounds which produce such units on hydrolysis. Some of the earlier known carbohydrates were sweet to taste, and in Greek the word saccharon means sugar. Thus the name saccharides was given. Examples of carbohydrates are sugar, glucose, starch and cellulose. Some more examples of carbohydrates in our daily life are: we clothe ourselves with cellulose in the form of cotton, linen, and rayon. We build furniture and houses from cellulose in the form of wood. Functions of carbohydrates. The carbohydrates are the chief energy providers. They are the constituents of bodily tissues. Classification. Carbohydrates can be classified on the basis of their behavior on hydrolysis. They can be divided into three categories: monosaccharides, oligosaccharides, polysaccharides. Monosaccharides. Monosaccharides cannot be hydrolyzed further to give simpler units of polyhydroxyaldehydes or ketones. On the basis of functional groups, they can be categorized into two groups. If a monosaccharide has an aldehyde group, it is known as aldose, and if the monosaccharide has a keto group, it is known as a ketose. Monosaccharides may contain 3 to 7 carbon atoms. Depending upon the number of carbon atoms present in a particular monosaccharide, they can be further divided into trioses, tetroses, pentoses, hexoses or heptoses. Let's have a look at some more examples of monosaccharides. Erythrose is an example of aldotetroses. Let's have a look at the structure of erythrose. Erythrulose is an example of ketotetroses. We are looking at the structure of erythrulose. Arabinose is an example of aldopentoses. It's time to see structure of Arabinose on the screen.
Students, let's have a look at the shown table on the screen. It shows the number of carbon atoms present in the parent chain, general term, aldehyde, and relevant ketone. If the number of carbon atoms is 3 in the parent chain, its general name is triose. The relevant aldehyde is named as aldotriose and the relevant ketone is named as ketotriose. If the number of carbon atoms is 4 in the parent chain, its general name is tetrose. The relevant aldehyde is named as aldotetrose and the ketone is named as ketotetrose. If the number of carbon atoms is 5 in the parent chain, its general name is pentose. The relevant aldehyde is named as aldopentose and the relevant ketone is named as ketopentose. If the number of carbon atoms is 6 in the parent chain, its general name is hexose. The relevant aldehyde is named as aldohexose and the relevant ketone is named as ketohexose. If the number of carbon atoms is 7 in the parent chain, its general name is heptose. The relevant aldehyde is named as aldoheptose and the ketone is named as ketoheptose.
Oligosaccharides. They can be hydrolyzed to give further simpler units of polyhydroxyaldehydes or ketones. Oligosaccharides on hydrolysis can yield 2 to 10 monosaccharides units. Obtained monosaccharides units may be same or different. On the basis of monosaccharides units released by them, they can be categorized as disaccharides, trisaccharides, and tetrasaccharides, etc. Disaccharides. On hydrolysis, disaccharides release 2 molecules of the same or different monosaccharides. Its examples are sucrose, maltose, lactose, etc. Let's have a look at the structures of sucrose, maltose, and lactose on the screen. Trisaccharides. On hydrolysis, these releases 3 molecules of the same or different monosaccharides. Its example is raffinose. The structure of raffinose is shown on the screen. Polysaccharides. On hydrolysis, polysaccharides yield a large number of monosaccharides units. These may be further classified as homopolysaccharides or heteropolysaccharides. Homopolysaccharides give only one kind of monosaccharides units on hydrolysis. Heteropolysaccharides give more than one kind of monosaccharides units on hydrolysis. Some common examples of carbohydrates are starch, cellulose, glycogen, gums, etc. ਕਾਰਬੋਹਾਈਡਰੇਟਸ ਨੂੰ ਕਲਾਸੀਫਾਈ ਕਰਨ ਦਾ ਇੱਕ ਹੋਰ ਵੀ ਮੈਥਡ ਹੈ। ਇਸ ਨੂੰ ਰਿਡਿਊਸਿੰਗ ਅਤੇ ਨਾਨ ਰਿਡਿਊਸਿੰਗ ਪ੍ਰੋਪਰਟੀਜ਼ ਰਾਹੀਂ ਸਪਸ਼ਟ ਕੀਤਾ ਜਾ ਸਕਦਾ ਹੈ। Reducing sugars, all those carbohydrates which reduce Felling solution and tolerance reagents are referred as reducing sugars. Reducing sugars include glucose, fructose, lactose, Arabinose and maltose. That is all monosaccharides, whether aldoses or ketoses. Non-reducing sugars, all those carbohydrates which do not reduce Felling solution and tolerance reagents are referred as non-reducing sugars. Example of non-reducing sugar is sucrose, and all polysaccharides like starch, cellulose, glycogen, dextrins, etc. In this part of the episode, we will study about glucose. It is a monomer unit of large carbohydrates like starch, cellulose, etc. It occurs both in free or combined form. It is present in sweet fruits and honey. Ripe grapes contain glucose in large amount, hence also known as grape sugar. It is an aldohexose. It is highly soluble in water, sparingly soluble in alcohol and insoluble in ether. Its solution is dextrorotatory, hence it is named as dextrose.
Let's have a look at the structure of glucose. It has 6 carbon atoms in it. It also contains 5 hydroxyl groups and 1 aldehyde group. Out of the 5 hydroxyl or OH groups, 1 is primary hydroxyl group and 4 are secondary hydroxyl groups.
Preparation of glucose. Glucose can be prepared by two methods. Let's discuss them one by one. From sucrose. Sucrose is boiled with dilute hydrochloric acid or sulfuric acid in alcoholic solution. Glucose and fructose are obtained in equal amounts. From starch. Commercially, glucose is obtained by hydrolysis of starch by boiling it with dilute sulfuric acid at 393 Kelvin under pressure 2 to 3 bars. This structure was assigned on the basis of these evidences. Evidences in favor of its structure. 1. Molecular formula. Its molecular formula comes out to be C6H12O6. Straight chain. When glucose is heated with hydrogen iodide, it forms N-hexane. It confirms presence of 6 carbon atoms in a straight chain. Presence of carbonyl group. Glucose reacts with hydroxylamine to form an oxime. It confirms the presence of carbonyl group. Moreover, glucose adds on to one molecule of hydrogen cyanide to form cyanohydrins. This again confirms the presence of carbonyl group. Fifth, presence of aldehyde group. Glucose on oxidation with bromine water forms gluconic acid. It confirms presence of carbonyl group as an aldehyde group because aldehydes on oxidation form carboxylic acids. Next point is presence of 5 hydroxyl functional groups. Glucose reacts with acetic anhydride, gives glucose pentacetate. It confirms presence of 5 hydroxyl functional groups in it. The next point is presence of one primary alcoholic group. On oxidation with nitric acid, both glucose and gluconic acid give the same dicarboxylic acid known as saccharic acid. It confirms presence of a primary alcoholic group in glucose.
ਵਿਦਿਆਰਥੀਓ, ਆਓ ਹੁਣ ਗਲਿਸਰਾਈਡਸ 'ਤੇ ਨਜ਼ਰ ਮਾਰੀਏ। ਇਸ ਵਿੱਚ ਇੱਕ asymmetric carbon ਹੁੰਦਾ ਹੈ। It means that 4 valencies of carbon atoms are satisfied by 4 different groups or atoms. It exists in 2 forms. These are D plus glyceraldehyde, that is with hydroxyl group on right, and L minus glyceraldehyde, that is with OH group on left. Now let's have a look at the structures of D plus glyceraldehyde and D plus glucose on the screen. Hence, glucose is correctly named as D plus glucose, where D represents the configuration and plus represents dextrorotatory nature of the molecule.
Cyclic nature of glucose. First point, glucose fails to give 2,4-dinitrophenyl test. Second point, it fails to give Skiff's test. Third point, it does not form hydrogen sulfide. Fourth point, the pentaacetate of glucose does not react with hydroxylamine, indicating the absence of free aldehyde group. Fifth point, moreover, glucose is found to exist in 2 crystalline forms, alpha form and beta form. Alpha form of glucose with melting point 419 Kelvin is obtained by crystallization from concentrated solution of glucose at 303 Kelvin. Beta form of glucose with melting point 423 Kelvin is obtained by crystallization from hot and saturated aqueous solution at 371 Kelvin. Due to the limitations already mentioned, the open chain structure of glucose was rejected. A cyclic structure known as hemiacetal structure was proposed.
In this structure, the aldehyde, that is CHO group, is involved in the formation of a ring with the OH group, that is the hydroxyl group, attached to the carbon 5. It is a 6-membered ring. The ring structure accounts for the 2 isomeric forms, alpha D positive glucose and beta D positive glucose structures are shown on the screen. These 2 cyclic forms exist in equilibrium with open chain structure. These 2 structures differ in the configuration of OH group, that is the hydroxyl group, and hydrogen atom attached to carbon 1 atom. Called as anomeric carbon. Such isomers are called anomers. These are shown on the screen.
Let's take an example to clear the concept.
The 6-membered cyclic structure of glucose is called Pyranose structure in analogy with pyran. Let's have a look at the structure shown on the screen. The first structure is of pyran, which is a cyclic compound with 1 oxygen and 5 carbon atoms in the ring. The first structure shown on the screen is of alpha D glucopyranose, and the second structure is of beta D glucopyranose. From the structures, it becomes clear that alpha D glucopyranose has the same stereochemistry at both C1 and C5 carbon atoms, whereas beta D glucopyranose has opposite stereochemistry at C1 compared to C5 carbon atoms.
Fructose. It is isomeric with glucose. It is found in honey and in many fruits. That's why it is known as fruit sugar or levulose. Let's have a look at the open chain structure of D- fructose on the screen. Molecular formula is C6H12O6. It has 6 carbon atoms in straight chain. Ketonic group is present at carbon atom number 2. It has D configuration. It is a levurotatory compound and hence named levulose. Thus, its name is appropriately written as D- fructose.
Fructose exists in two cyclic forms, alpha D fructofuranose, beta D fructofuranose, which are obtained by the addition of hydroxyl group at carbon 5 to the carbonyl group. The ring thus formed is a 5-membered ring and is named as furanose with the analogy to the compound furan. Let's have a look at the structure of furan shown on the screen. It is a 5-membered cyclic compound with 1 oxygen and 4 carbon atoms. Now, let's see the next structure. It is alpha D fructofuranose. In this structure, 6 carbon atoms are present in a straight chain. These are numbered as 1, 2, 3, 4, 5 and 6. 5 hydroxyl functional groups are present at C1, C2, C3, C4 and C6 carbon atoms. 1 keto group is present between C2 and C5 carbon atoms. Now see the last structure. It is beta D fructofuranose. In it, 6 carbon atoms are present in a straight chain. These are numbered as 1, 2, 3, 4, 5 and 6. 5 hydroxyl functional groups are present at C1, C2, C3, C4 and C6 carbon atoms. 1 keto group is present between C2 and C5 carbon atoms. What is the difference between alpha D fructofuranose and beta D fructofuranose? Difference between them is, in the case of alpha D fructofuranose, OH group attached to carbon 2 is present on right hand side, whereas in case of beta D fructofuranose, OH group attached to carbon 2 is present on the left hand side. It can be well understood by drawing Howard structures of them. Howard structures of alpha D fructofuranose and beta D fructofuranose are shown on the screen.
Preparation of fructose. Students, as we have studied that on hydrolysis, disaccharides yield 2 molecules of same or different monosaccharide. Thus, fructose is prepared by hydrolysis of sucrose. Sucrose reacts with water to form D plus glucose and D minus fructose.
Let's look at a table showing comparison between glucose and fructose. First, let's discuss similarities. Both react with acetic anhydrides to form pentaacetyl derivative. Both react with methyl alcohol and hydrochloric acid to form methyl glucoside. Both react with hydrogen cyanide to form cyanohydrine. Both react with hydroxylamine to form oximes. Both react with phenylhydrazine to form glucosazone and osazone respectively. Both on fermentation with yeast form ethyl alcohol and carbon dioxide. Both reduce Fehling solution and ammoniacal silver nitrate, that is Tollens reagent. Now let's discuss points of differences in them. Glucose is dextrorotatory, whereas fructose is levorotatory. Glucose reacts with lime to form calcium glucosate, which is soluble in water, whereas fructose reacts with lime to form calcium fructosate, which is insoluble in water. Glucose on reduction forms sorbitol, whereas fructose on reduction forms sorbitol and mannitol. Glucose on oxidation forms gluconic acid, whereas fructose on oxidation forms glycolic acid and tartaric acid. Glucose reacts with sodium hydroxide to form brown resin mass, whereas fructose does not react with sodium hydroxide solution.
Sucrose. Sucrose is commonly known as table sugar. It is a disaccharide of glucose and fructose. Its molecular formula is C12H22O11. It is formed by plants only and not by other organisms.
We are looking at an image showing dehydration synthesis of sucrose. Glucose and fructose combine together to form sucrose and one molecule of water. Hydrogen, that is H from glucose, and hydroxyl, that is OH from fructose, is shown on the screen to form water molecule. Let's have a look at the structure of sucrose showing alpha D glucose and beta D fructose. These two structures are joined together by an oxide linkage after losing one water molecule. This oxide linkage is also known as glycosidic linkage. Let's study the process of formation of sucrose in detail. In case of alpha D glucose, 6 carbon atoms are present, and in case of beta D fructose, also 6 carbon atoms are present. Carbon present at position 1 of alpha D glucose links itself with position 2 carbon atom of beta D fructose. As a result, sucrose is formed. An important point to be noted about sucrose is that it is a non-reducing sugar because the reducing groups of glucose and fructose are involved in glycosidic bond formation.
Next comes maltose. Let's have a look at the Howard structure of maltose. It is formed with the help of two alpha D glucose units in which carbon 1 of one molecule of glucose is linked to carbon 4 of another glucose molecule. Thus, free aldehyde group can be produced at carbon 1 of second glucose in solution. Hence, maltose is a reducing sugar. Lactose. Next among disaccharides is lactose. It is more commonly known as milk sugar since it is found in milk.
Let's have a look at the Howard structure of lactose. It is formed with the help of beta D galactose and beta D glucose. Here in this molecule, the linkage is between carbon 1 of beta D galactose and carbon 4 of beta D glucose molecule.
It is again reducing sugar because of the occurrence of free aldehyde group at carbon 1 of the beta D glucose in solution. Starch. Starch is the main storage polysaccharide of the plants. It is found in potatoes, wheat, maize, rice, and barley. Thus, it is the most important dietary source for human beings. An interesting fact related to starch is that no two sources give identical starch.
On hydrolysis with dilute acids or enzyme, starch breaks down into molecules of variable complexity, and finally maltose and D-glucose are formed.
Starch does not reduce Fehling solution. It does not reduce to Tollens reagent. It also does not form an ozone. It indicates that all the hemiacetyl hydroxyl group of glucose units at carbon 1 are linked with glycosidic linkages. Composition of starch. Starch is made up of two polysaccharide components. They are amylose and amylopectin.
Let's have a look at the structure of amylose. These units are joined together by a 1,4-glycosidic linkage involving C1 of the glucose unit and C4 of another glucose unit. Amylose. Amylose constitutes about 15% to 20% of starch. It is water soluble and has long unbranched linear chain. It has approximately 200 to 1000 D-glucose units. Its molecular weight can range from 10,000 to 5 lakhs. Amylose gives blue color with iodine. Amylopectin. Amylopectin constitutes about 80% to 85% of starch. It is water insoluble and has branched chain with 20 to 30 glucose units per branch. These units are held with two types of glycosidic bonds, a 1,6 glycosidic bond at branching points and a 1,4 bond in the linear chain. Amylopectin does not give blue color with iodine.
Cellulose. The next one is cellulose, which is the most abundant organic substance in the plant kingdom. It is the chief constituent of the cell walls of bacteria and plants. Plant walls contain about 45 to 50% cellulose. Now it's time to view structure of cellulose.
It is large linear structure and its individual strands align with each other from beginning to end by multiple hydrogen bonds. This type of arrangement provides rigidity in its structure. Because of this nature, it is used effectively as a cell wall material.
Cellulose is composed of beta D glucose units linked by beta 1 to 4 glycosidic bonds. It is straight chain polysaccharide which on hydrolysis yields beta D glucose. Due to the lack of an enzyme that can cleave beta glycosidic bonds, all mammals cannot digest cellulose. Large population of bacteria present in the stomach of ruminant mammals like cattle, sheep, etc. breaks down the cellulose with the help of enzyme cellulase. It is then digested and converted into glucose. Now come to chemical properties of cellulose. Cellulose neither reduces Tollens reagent nor Felling's solution. It is not hydrolyzed as easily as starch, but on heating with dilute sulfuric acid under pressure yields only D-glucose. It also does not form osazone and it is not fermented by yeast. Importance of carbohydrates. In the end, the importance of carbohydrates is summed up as follows. First, in living systems, these act like biofuels, that is, to provide energy for functioning of living systems. Next is, these act as reserve food material in the form of starch in plants and as glycogen in animals. Next, these are essential components of biomolecules like RNA and DNA. Next, these act as the chief structural material of cell wall of bacteria and plants, that is, cellulose. Next point is, carbohydrates provide raw materials for many industries like textiles, paper and lacquers and breweries. Another point is, these are found in biosystems in combination with many proteins and lipids. Next, cellulose in the form of wood is used to build furniture and houses. ਬੱਚਿਓ, ਮੈਨੂੰ ਉਮੀਦ ਹੈ ਕਿ ਤੁਹਾਨੂੰ ਅੱਜ ਦਾ ਇਹ ਪਾਠ ਕਾਫੀ ਦਿਲਚਸਪ ਲੱਗਿਆ ਹੋਵੇਗਾ। ਥੈਂਕ ਯੂ ਫॉर योर ਅਟੈਂਸ਼ਨ। ਅਗਲੀ ਵਾਰ ਇੱਕ ਹੋਰ ਨਵੇਂ ਵਿਸ਼ੇ ਦੇ ਨਾਲ ਮਿਲਾਂਗੇ। ਤਦ ਤੱਕ ਲਈ ਇਜਾਜ਼ਤ।